Hello, I'm Roger, and this summer I worked with Charles Olmsted at the UW-Madison McMahon Lab on a project about extracellular electron transfer. So before I get into it, just a little bit about myself. I really love bacterial films. Here's a picture of me with my favorite, which is called Leptothrix discophora. Um, I am a rising senior at Oberlin College, currently an undergrad. I'm double majoring in both biology and environmental studies. And this is my second year at Trout Lake, second year in quotations because unfortunately I am working remotely from Connecticut currently. So now to get right into it, I'm sure a lot of people are wondering what exactly extracellular electron transfer is, and I definitely was when I started this summer project. So the way that I like to think about it is that it's actually very similar to something that human beings already do in their cells. So to generate energy, we actually have electrons in our bodies flow along something called the electron transport chain that's present in a lot of our cells. And when these electrons get to the end of the chain, they have to go somewhere. So as humans, as animals, we put them onto atmospheric oxygen. And this atmospheric oxygen comes from the environment, the air that we breathe. And this is the reason why breathing is so important. It basically keeps us alive because it's needed to generate energy um, via this method. So bacteria that are capable of performing EET actually do something very, very similar. But instead of using atmospheric oxygen, they extend the electron transport chain outside of their own cells so that they can interact with the surrounding environment. So here is the interior of the cell, electron flowing out and being put onto this iron molecule outside in the environment. So bacteria that can perform EET can do so with stuff, ambient materials in the environment, such as iron, but this also includes dissolved organic matter. So what exactly are we talking about when we talk about dissolved organic matter? And specifically in this presentation, we're going to be talking about color dissolved organic matter, which is a type of dissolved organic matter that is named because it gives bog water its very rich brown amber color. If you've ever seen bog water, it's very, very this deep brown amber orange color. And here's an example to the right of what a color dissolved organic matter molecule might look like. So bogs are especially high in this type of material, and some bacteria can use this for EET and avoid the cost of transporting the molecules inside and outside the cell, because if you recall, they're able to interact with it directly outside in its environment. So our research question is one of the factors that determines the prevalence of how much EET actually happens in bogs the quantity of dissolved organic matter in the bog, or is it some other factor? So to look into this, we and our collaborators collected a bunch of bacterial samples from lakes in the following areas. And my job specifically this summer was to calculate the percent of bacterial species in each lake that perform EET. And this was called the EET ratio. It was the bacterial species that do EET over the total number of bacterial species found in each sample. And when that was graphed against dissolved organic carbon, very similar to dissolved organic matter, this is the resultant graph. So you can see the blue spots are some lakes that you might know. The names might be familiar to you because they're here, home in Wisconsin. And this trend line describes the relationship of the graph. As you can see, as dissolved organic carbon content increases, so does the EET ratio percent. So as there is more dissolved organic carbon in any specific lake, the trend is that EET ratio percent is also higher. There is more EET going on in these lakes. So this is good because it definitely supports our hypothesis, our initial hypothesis going into this, which was that dissolved organic matter was one of the controlling factors for how much EET happens in a lake. But we also looked into some other things. So in addition, total iron, as iron amount increases, we also saw that EET ratio would increase. In the case of dissolved oxygen, we actually saw the opposite. When dissolved oxygen increases, EET ratio decreases. But this makes sense. We were expecting this as well because a lot of bacteria that perform EET do so in anoxic environments, environments without any or have very, very little oxygen in them. So for next steps for the rest of the summer, we're going to be looking at something very similar, but we're going to be looking into sulfate levels in the lakes, how pH, acidity, and or basicness of the water might affect EET ratio levels, and the presence and amount of methane. So one of the major motivations for researching EET actually has to do with climate change, surprisingly enough. EET might play a pretty big role in climate change. And the way that it might do so is as CO2 and methane drive climate change, they contribute to the greenhouse effect, which contributes to global warming. And this global warming makes permafrost in places like Canada up north melt even faster. So as this permafrost melts, it melts down into these melt ponds that you can see here. This is a picture of Canada, of a site in Canada. And bacteria can then proliferate once these melt ponds have been created. Based on ours and other studies, around half of the organisms in these ponds contain EET genes and are likely capable of doing EET. And because EET likely influences both the production of CO2 and methane by bacteria, by understanding exactly how EET works and how it affects these things, we're going to be able to better predict how the melting permafrost will ultimately affect climate change. One theory or assumption is that as this melting progresses and gets worse, um, production of CO2 and methane might actually hasten it and make it go even faster than before. But of course, we'd have to look more into stuff like this first. So that's one of the things that we are thinking. So I would like to thank everybody for listening. I would really like to thank Jack and Pat Lane and the rest of the Lane family for providing funding. I'm extremely grateful this year. Um, Without their generosity, I would not be employed this year due to coronavirus. Um, so it really means the world to me. And I would also like to thank my coworkers, Charles Olmsted, for helping me out whenever I didn't know how to do something in R, everyone at the McMahon Lab, and then everyone at Trout Lake Station, of course. And then additional data was sourced from the people listed below. This is where we got much of our collaborator data from. Thank you so much for listening. Bye.